Good morning, students. We are going to continue with chapter 2 of NCERT, that is sexual reproduction in flowering plants. Students, we have covered the process of pollination along with its various agents in our previous presentation. Now it's time to study how fertilization is being carried out and ultimately embryo is formed. So let's continue with the presentation. Topics which are being covered in this tutorial are pollen-pistil interaction, artificial hybridization, double fertilization, post-fertilization events, endosperm, in which we have to discuss about its development and functions. Then finally, the structure of dicot embryo. First of all, we have to focus on pollen-pistil interaction. For better understanding, refer this diagram. Here you can see many pollens landed over the stigma for the fertilization, but only one pollen is being successful to reach towards the embryo sac. So recall, pollination does not guarantee the transfer of the right type of pollen, that is compatible pollen of same species as the stigma. Often pollen of wrong type, either from other species or from the same plant. If it is self-incompatible, also land on the stigma. The pistil has the ability to recognize the pollen, whether it is of compatible type or incompatible one. If the compatible pollen land over the pistil, it promotes post-pollination events that bring about fertilization. If the pollen is of incompatible type, the pistils simply reject that pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by the acceptance or rejection is the result of continuous dialogues between pollen grain and the pistil. This dialogue is mediated by the chemical component of the pollen interacting with those of the pistil. You can easily recognize some of the pollens are able to germinate but do not go beyond the style because they are of incompatible type, and only the compatible pollen is successful to reach towards the embryo sac. So you can easily see the chemicals which are being present over the stigma go for the continuous dialogue with the chemicals of the pollen to know whether they are of compatible type or incompatible one. So this part is quite clear to you. Now move to the other diagram. This pollen structure is quite familiar to you, and this is two-celled pollen. And you can recall, if the pollen are going to be shed at two cell stage, then the generative nucleus is going to divide by the process of mitosis to form two male gamete. However, the vegetative cell helps in the formation of pollen tube. So the male gamete help in the fertilization. However, the vegetative nucleus helps in the formation of pollen tube from germ pore. Now, let's look the important of the key points of the pollen pistil interaction. All the events from the pollen deposition on the stigma until the entry of the pollen tube into the ovule are together called pollen pistil interaction. It is quite a dynamic process in which pollen of compatible type are going to be recognized by the chemicals being present over the stigma. Only compatible pollens are being encouraged by the pistil for the growth and development of the pollen tubes. Pollen tube growth through the stigma and style reach to the ovary through the micropylar and reaches the synergids, and ultimately fertilization going to take place. Now, not only the natural pollination process is helpful for the fertilization, sometimes we also want desirable characteristic of our importance into that of the plant. So what kind of technique we are going to use? That is the artificial hybridization. In this, we have to follow two methods, that is emasculation and bagging, for successful achievement of desirable characteristics in the plant. As you can see, emasculation is the removal of pollen before any sort of dehiscence in bisexual flowers. And then the flower is going to be covered with the help of paper bag or any polythene bag with some perforation pores for the gases exchange to prevent contamination from unwanted pollens. On attaining maturity, the mature pollens from the desirable plants are going to be dusted on the stigma and the flower going to be rebagged. You can understand emasculation and bagging with the help of the diagram. The anther are going to be removed with the help of scissor or forceps, and these anther can be used to dust pollen of desirable type on another flower as well. And here you can see the emasculated flower is being bagged with the help of cloth bag or paper bag, and one tag has been put to know about the date of emasculation. Now let's move to the fertilization event. In angiosperm, double fertilization along with triple fusion used to take place. So first of all, we have to understand the two male gamete being formed into the pollen are going to be utilized for the fertilization event. And one of the male gamete going to fuse with the egg to form the zygote. However, the other male gamete fuses with the polar nuclei to form pen. And what is pen? That is primary endosperm nucleus. And this is referred as triple fusion. Why it is referred as triple fusion? As you can see that two nucleus of the central cell going to be fused with one nucleus of the male gamete, and ultimately three nucleus are going to be fused with each other. That's why it is referred as triple fusion. As syngamy, that is fertilization of the male gamete with the egg, this is referred as syngamy. And triple fusion going to take place simultaneously in the embryo sac, this is referred as double fertilization. And central cell after triple fusion forms primary endosperm cell or the primary endosperm nucleus, which later develop into the endosperm. The zygote going to develop into the embryo. So let's understand the whole process. That pollen tube enters through that of the synergids, and male nucleus going to be fused with the egg to form the zygote, and one male nucleus going to fuse with the two nuclei of the central cell, that is polar nuclei, and triple fusion going to take place here, and this triploid nucleus ultimately going to form primary endosperm cell or the pen, that is primary endosperm nucleus. And this zygote going to be converted into the embryo. Stage is the globular embryo, then heart-shaped embryo. With the heart-shaped embryo, the cotyledon number is quite clear to you. Yes, these are the two, so the dicotyledonous seed is going to be formed later on. Okay, and you can see two cotyledons here also in the mature embryo. So this is the development of dicot embryo. Now we have to study about the post-fertilization events. These are four in number. First of all, the development of pen, that is primary endosperm nucleus. And second is development of embryo from zygote. Third is development of seed from ovule. And finally, the fruit formation from the ovary. Now, first of all, we have to understand the endosperm development. This is very important, followed by embryo formation. This develops from pen, that is primary endosperm nucleus. Pen undergoes successful nuclear division to give rise to free nuclei, and this stage of endosperm development is called free nuclear endosperm, because there is no cell wall formation till this stage, and later the cell wall formation going to take place from the periphery towards the center, and formation of cellular endosperm going to be achieved thereafter. Functions of endosperm. This is very very important from the nourishment point of view. The endosperm is going to provide all sort of nourishment to the developing embryo. As you can find out in coconut, the water represent the liquid endosperm. Okay, and liquid endosperm is also referred as free nuclear endosperm. And the white kernel portion which we usually eat is the outer part, and it is multicellular endosperm. The cellular form is being achieved into the white kernel stage. Sometimes endosperm is going to be completely consumed by the developing embryo, as in the case of peas, beans, that is Fabaceae family. And in some of the seed, it is going to be persist up to the end, that is in the case of coconut. Embryo development. This is very, very important. The early development of the embryo is called as embryogeny. Embryo develops from zygote at the micropylar end of the embryo sac. The all sort of nourishment is going to be provided by endosperm. The zygote divides mitotically and form proembryo. And in dicot, we have studied that globular embryo, heart-shaped embryo, and finally the mature embryo with cotyledonous distinction has been achieved by the repeated mitotic division. So, how the dicotyledonous embryo looks like? It usually have embryonal axis along with two cotyledons. The portion of embryonal axis above the level of cotyledon is referred as epicotyle, which is going to form shoot system later on. And the cylindrical portion below the level of cotyledon is referred as hypocotyle, which is going to form the root system. Okay, sometimes the root tip is often covered with the root cap, which is referred as calyptra. Now, have a look over the dicot embryo. You can easily see the plumule section, which is a future shoot, and this is the radical portion, which is going to form the future shoot root system. And this is hypocotyle region, and two cotyledons are quite distinct in this diagram. That's all for today. Thank you.